We find ourselves back on the ramparts to discuss our next topic, macrophages. And it's a fitting choice since macrophages are the sentinels or the night watchmen of the immune system. They patrol our bodies on the lookout for invaders. Hopefully by now you remember that our recurring symbol for the macrophage is this macrocage guy. But we're going to get a little more detailed in this lesson, and in fact there are two types of macrophages. Those that are descended from monocytes, and those that aren't. This first guy we've drawn in is wearing a monocle to symbolize that he was directly derived from a monocyte. These monocyte-derived macrophages can be distinguished from non-monocyte-derived macrophages by their CD14 cell surface marker. And so we'll have him marking out a fortnight here. Get it? 14 is 2 weeks, which is a fortnight. See, we don't just teach you science here at Sketchy. As opposed to monocyte-derived macrophages, non-monocyte-derived macrophages are derived from embryological structures. So we've drawn in a baby-faced macrophager without a monocle to remind you of this. Some examples of these types of macrophages are Kupffer cells in the liver and alveolar macrophages in the lungs, which we've denoted with this little man's lung vest and liver bag. Oh, and looks like they got into a little tiff, probably over some key differences between them. Our monocle-wearing macrophager is holding up a bloody tissue to his nose to remind you that monocyte-derived macrophages circulate as monocytes in the blood and then get recruited to sites of injury or inflammation, where they transform into macrophages once they've arrived in the tissue. That's in contrast to non-monocyte-derived macrophages, which arise and live out their days solely in tissues. It's time to hug it out, you two, and remember what you have in common. And what they do have in common is that they both do all their important work in tissue, regardless of where they came from. So now that we've mentioned it, let's talk about this important work that macrophages do. Depending on different levels of activation, they can have different functions. At rest, macrophages patrol the tissues and act like garbage collectors. They clean up the cellular debris of dead cells and generally just keep everything tidy, which is why these two have been storing all the castle's garbage in their cages. Nice. Gross, but nice. Now, you may be wondering what exactly activates a macrophage. And you're in luck, because our 4 helper squire and natural killer assassin, who represent helper T cells and natural killer cells respectively, are running in with their interfering ginger ferrets, our symbol for interferon gamma, to do just that activation. This memorable scene is here to sear into your brain that macrophages are activated by interferon gamma, which is secreted by helper T cells and natural killer cells who have encountered a pathogen. And so, now what? What happens when macrophages get activated? Well, it's quite a sight to behold. Interferon gamma's effect on a resting macrophage is to cause it to express more class II MHC molecules, which essentially makes it a better antigen-presenting cell, and that's why this macrophager is presenting the antigenic dragon's tooth to the helper T squire, since macrophages often present antigen back to helper T cells to activate more of them. This job is crucial in the innate immune response because the more antigen presented, the more the immune system sees and can react to. But macrophages don't just want to present things, they want to destroy them too. Remember, they're phagocytes. While our macrophages are on patrol, they can run across bacterial molecules like lipopolysaccharide, which are an example of a PAMP, or pathogen-associated molecular pattern, which we've symbolized with this pamplemousse, obviously. When they see those molecules, macrophages get extra activated to eat the enemy, aka total destruction. So, importantly, these activated macrophages secrete TNF-alpha, represented by these alpha-shaped tuna fish getting knocked over and released when our macrophage takes a chomp into their juicy pamplemousse. TNF-alpha is a powerful cytokine that you might have heard about as an important mediator of sepsis. That's because it recruits other immune cells to the site of infection, just like the first responder neutrophils rushing toward those flopping tuna fish. Now that's unforgettable and probably smells terrible. But superactivated macrophages don't stop there. Once they recognize a pathogen, they also increase production of reactive oxygen species, like hydrogen peroxide and nitric oxide, both of which can directly kill invaders. We've drawn in a bottle of peroxide and a no running sign to represent both of these things, since, that's what you'd normally see if there was a giant tuna fish spill. And finally, macrophages don't just destroy things, they help the body heal as well. Certain cytokines, such as IL-4 and IL-13, though don't worry about memorizing those numbers right now, cause macrophages to promote the healing of damaged tissues. They do that by releasing substances that cause new blood vessel growth, aka angiogenesis, and by laying down a collagen-rich extracellular matrix to help with fibrosis. These fibrous patches and red vines on the castle walls are a reminder to you that macrophages work hard to keep this kingdom intact. Let's do a quick review before we take a nap up here on the wall. Macrophages are phagocytic cells of the innate immune system that reside in tissues. They can either be non-monocyte derived, which means they arose in tissues from embryological structures, or monocyte derived, which means monocytes in the blood migrated into tissues where they differentiated into macrophages. Monocyte-derived macrophages can be distinguished by having the CD14 cell surface marker. Macrophages have many functions and levels of activation. At rest, they act like garbage collectors, cleaning up cellular debris and targeted pathogens via phagocytosis. If they're activated by interferon gamma, which is released by helper T cells and natural killer cells, then macrophages go on to become a little more active. Interferon gamma causes upregulation of MHC2 molecules, which means the macrophage becomes a very good antigen-presenting cell. A macrophage can also directly recognize a pathogen, and if it does, it can destroy that pathogen via phagocytosis, and also secrete TNF-alpha, which is an important mediator of inflammation. These extra activated macrophages also produce reactive oxygen species and nitric oxide, which can both directly kill pathogens. And finally, macrophages also promote angiogenesis and fibrosis within damaged tissues, which means they can help with healing after the battle has been fought. And that's it for our patrolling macrophages. Stay tuned for a video on first responders, but first, lunch. Grapefruit and tuna fish sandwich, anyone? No? Really? All right, more for me.